Okay, so good afternoon, everybody. The following half an hour we will spend together with mass spectrometry, and I will make you familiar with different modes of operation of this method. We will start with the simplest one, which is just to check the sample quality, to confirm the identity and purity of your protein samples. Later on, we will go to more supervised modes of mass spectrometry. If you want to check the quality of your sample, the first method of choice you are going to use is Molditov mass spectrometry. So in the first part, I will try to explain you how this method works, because I know that some people don't like this method. For example, if you speak Spanish or you are from a Spanish-speaking country, the word maldito means something very bad. Yeah? But the other people don't like Molditov mass spectrometry because of its very complicated name. In contrast to the name of the method, to make the analysis is something very easy and can be accomplished in a few minutes. If you have your protein molecule and you want to determine its molecular weight, you simply mix it with an excess of a so-called matrix, which is usually a small molecule derived from benzoic, salicylic, or cinnamic acid, and you allow to dry this mixture of your protein sample with the matrix on a metallic support. After being dried and introduced to the mass spectrometer, you just apply laser pulse on that mixture, and what you obtain is entire molecule of the protein released into the gas phase in a form of an ion. Nobody knows what exactly happens during this ionization process. It has not been fully understood on the physical-chemical level, so it is somewhat an alchemy. Nevertheless, we know that it works, and whenever we obtained enough stable ion of the protein, then it is very easy to determine its molecular weight on the basis of its behavior under electrical or magnetic field. In the case of time of flight mass spectrometry, it is electrical field. Simply the time of flight is dependent of the molecular weight and of the charge number of the molecule. A result of this analysis is a mass spectrum, where in the case that we had a pure protein, we obtain simple mass spectra where all signals can be assigned to different ionization form of your molecule. The mass accuracy of TOF analyzers of larger proteins is about 500 ppm. Detection limit is quite nice. So whenever you obtain this peak after a single sample preparation, which takes some 2 minutes, if the mass matches your protein, there is high probability that it is really your protein, because I think it would be really a shame if you purify a different protein of the same mass. If you obtain such a result, then you may see your protein as a minority signal. It means probably that you should prepare a new fresh one, because this one has undergone some degradation. Or if you look in closer detail, you may find some modification of the protein. For example, here, this protein was treated by a kinase, so some phosphorylation was expected, and this rapid intact mass analysis can reveal you this kind of modification by detecting a distinct peak with a mass shift corresponding to phosphorylation. You can also study the degree of phosphorylation. You can see that now the second peak corresponding to the phosphorylated form of the protein is more abundant. However, without the previous knowledge on the kinase treatment, these signals simply cannot tell you that it is truly a phosphorylation. But we had some background which can lead us to this conclusion. In the following example, we will show you some episode from our core facility live, when our customer brought us a protein sample to check its identity. We obtained this sequence from the customer. There were 2 samples, the concentration is here to distinguish between them. They had 2 names based on the concentration. The calculated mass of the protein was this one. However, when we look at the result of the mass spectrometric analysis, both samples showed a different value. And so we sent this result to the customer, asking him what could be the clue, what might have happened. So here is the answer. So the customer just sent us information that there are 2 more amino acids that he didn't tell us. However, if we go back to the analysis, the mass difference is higher than could be accomplished by any 2 amino acids. So we asked the customer in the following email which 2 amino acids were added to the protein. The result was surprising. There were not 2, there were 4 other amino acids. And now the theoretical mass nicely matches one of the samples. But what could happen to the second one? So we could deduce that it could be some other modification, some uncleaved tag, or could be a totally different protein. For that purpose, we must go to analyze the sample in a more sophisticated way. Routinely, we digest it by some protease. The first protease of choice is always trypsin, which cuts the protein into peptides, and the subsequent mass spectrometric analysis will reveal the molecular weights of the peptides. What is interesting, now we go to much better mass accuracy of tens of ppm, and also the detection limit is much lower. For that reason, if you want to identify a protein, usually it is carried out after the digestion, because then mass spectrometry is really revealing better detection limits. This so-called peptide map is not a reliable platform for protein identification. We must go on and carry out fragmentation of individual peptides from this peptide mixture obtained by proteolysis of the original protein. For that purpose, we can also use mass spectrometry, also Marlitov mass spectrometry, where we, for example, select this ion for fragmentation, and then we apply higher laser power on the sample so that the peptide gets not only ionized, but also its ions are metastable and undergo fragmentation during the flight in the analyzer. However, if we fragment all the peptides from the entire peptide map, it would be impossible to interpret the MS-MS data. For that reason, we must add here some small slides with added voltage. It is called ion gate, and here you have it in detail. As high voltage is applied on one of the slides, all ions that are passing through are deflected and their life is finished here. However, exactly at the time when our ion of interest is passing, we switch off the voltage and only this peptide and its fragments can be detected. Fortunately, during MS analysis, peptidic bond gets fragmented. So from this model peptide composed of 6 amino acids, we obtained by MS analysis, by fragmentation within the mass spec, some shorter peptides which correspond to fragments containing either the C- or N- of the peptide. And by simple mathematical calculations, you can find that the distances between the fragments we obtained by MS correspond to individual peptides in the peptide sequence. However, there are some exceptions. In this case, we didn't find any reasonable difference between the masses. Even the principal fragment that can be found in every MS data obtained from tryptic peptide, when they are finished by arginine or lysine, you always see some of these 2 signals, but here they are missing. Instead of 175, we have 179, and then we obtained expected response from the customer. He didn't tell us that his protein is N15 labeled. He supposed that this information was not important for us. So finally, we can go back to intact mass analysis, where when we calculate the molecular weight of N15 labeled protein, we see that it is in perfect agreement with our analysis. So this is just to demonstrate to you what we can do in case when the intact mass analysis seems to fail, but I think the communication with the customer is the principal thing that can save you much time. So now we will leave the simplest setup of mass spectrometry, and I will try to show you in the following section what deeper insight can be provided to you by mass spectrometry-based proteomics. So I will have 2 examples of analysis, but we will start again with Marlitov MS. If you analyze bacterial cells by malignant of MS directly, that you smear them on the sampling target, overlay with the MOLDI matrix, and just carry out the analysis. You obtain a protein fingerprint that can be used for the identification of the bacteria, which is a method now widely used in routine clinical diagnostics. But I'm showing this feature for opposite reason. This is also the weak point of malignant of MS, because the bacterial cells contain some thousand of protein. However, by their profiling by malignant of MS, we see some 50 signals. Due to signal suppression effects, only the most abundant, the low molecular weight and basic proteins can be detected directly from complex protein mixtures. If your protein of interest doesn't fulfill that criteria, it will simply not be detected. And usually all abundant proteins have been discovered and no one is interested in them. So for the purpose of their detection and studying by mass spectrometry, you must couple the method to some separation step. For that purpose, we established we just applied several combinations of mass spectrometry coupled to liquid chromatography. This is the picture of our crowded laboratory. Maybe you would be curious, why do we need so many machines? You might guess this, as all analytical chemists like to have a lot of toys to play. Maybe you are right. Also, time by time, some of the mass specs is out of order, so we need a substitute. That's also a reason. But the main reason is that each of these LC-MS system is able to reveal another feature of the sample, can respond to another type of question that you pose to the sample. For example, this mass spectrometer based on ion trapping, 10 years ago it was the best mass spec we had, now it is the weakest one, and now it is just used for checking the digest quality. This is a QTOF, which is excellent for larger peptides. This is a triple quad for targeted proteomics. That means that you know what you're looking for, so you tune the quadruples in the mass spectrometer to obtain the signal of the peptide of your interest with the highest quality, and you can then quantify it. And this is the new generation of mass spectrometers based on Orbitraps. And not only it is a special kind of FT-based analyzer, but also inside the instrument you can find many features, many parts of ion optics, so you can play with the ions based on the properties that are desired. For example, for special kinds of post-translational modification, different setups of MS-MS fragmentations are needed. And it is everything available here. You can choose how the ions from the moment when you ionize them by electrospray will be treated with the aim to obtain maximum information desired from the system. As too many samples arrive, we needed another Orbitrap, because with the time the quality of the ion optics is improving, so this instrument is faster and more sensitive. And we found that two are not enough. We have also another Orbitrap instrument recently installed, which hasn't got so rich ion optics, but it is really very fast. It is excellent for large-scale studies where in a single LC-MS run, it can identify and quantify over 1,000 of proteins. To see what we can do with this instrument, I selected two examples of recent studies conducted in our laboratory. So, typical Czech sample, a beer, was desalted, and then it was treated as a common proteomic sample. So, proteolysis was carried out on a filter, which is a very smart technique for desalting, for purifying. Simply, it is a molecular weight cutoff filter that you centrifugate, and always you add the reagent or some purification agent, and finally you obtain a digest fully compatible with MS analysis. However, we also tried another methodical setup where we involved isoelectric focusing of digested peptides of beer, and then the strip of IAF was cut into pieces and the peptides were extracted, and each fraction was again analyzed by LC-MS with Orbitrap mass spectrometer. Here is the result. So overall, we were able to identify almost 2,000 of proteins in beer. And you can see how it is important to develop method variants and to improve the separation efficiency, because using the routine protocol, we saw only about 600 proteins, but as analytical chemists, we wanted to see more, and by employing the fractionization, which was a method optimized in our lab, we can reach the final result. In fact, this is not the final result, and every result should undergo some bioinformatic treatment. There is just an example. These are epitopes. These amino acid sequences are responsible for allergic response of people suffering from celiac disease. And unfortunately to them, you can see that several proteins in beer, about 55 which we detected, contain these epitopes. So the result is obvious. That's the reason why a beer in its crude form cannot be served to people suffering from this disease. Another example will be based on study of post-translational modification. As we are a group aimed mostly at plant research, we always study this boring Arabidopsis thaliana. I don't understand why plant biologists chose such a stupid plant, there is nothing to eat on it and no nice flowers, nothing, but they always bring us this. So you can see that our colleagues wanted to study post-translational modifications in histones. Again, it is not a straightforward task that you would put a vial in the mass spec and you run automatic analysis. You can see that there are 7 steps of extraction, derivatization. Each of them had to be optimized to obtain the final result. This is just a scheme where you can see how massively the histones can be post-translationally modified, and also we were able that to quantify these modifications after some treatment of inhibitors of acetylation. So this is just an overview what we can do, but we have to combine analytical chemistry, separation methods, and the state-of-the-art mass spectrometric tools. So now let's go to the last part of my talk. Maybe some of you are interested in structural biology. Is there anybody? Yeah, there are 3 people. Great. So there is reason to go on. So just a brief overview on the now most popular techniques that are used in this field and where you can employ mass spectrometry. So I have selected 3 of them. The first one is called hydrogen deuterium exchange. Principally, it is something very simple. Just when your protein is exposed to the deuterated solvent, the hydrogens that are accessible to the outer part of the molecule that are not fixed in some firm structure within the protein are being exchanged. And of course, this exchange is changing the molecular weight of the protein and of the peptide parts so that we can detect it by mass spectrometry. The setup of this principally very simple method is quite complicated because the exchange, even stopped by low pH, still carries on. So to obtain some relevant data, the proteolysis must be optimized. Pepsin is used instead of trypsin because it is very rapid and it can act at low temperatures. What you obtain are, again, proteolytic peptides, which can be very fast separated by LC and detected by MS. What you obtained are differentially labeled proteins or peptides. Unfortunately, data treatment is quite complicated because their signals overlap, and also the chromatographic behavior of light and heavy forms of the peptides is different, so the forms do not commigrate perfectly. Usually, this experiment is carried out in differential form, so you have some protein with or without presence of a ligand, and you see how the conformation of the protein changed, and then you see difference also on the level of mass spectra of the light and heavy forms. So another method is called protein surface labeling. If you expose a protein in a solution to some reagent, it is expected that it will bind only to the surface of the protein. So that if you then digest that protein to peptides and you analyze them by LC-MS, you simply can deduce which part of the protein complex was exposed to the solvent and which was hidden inside. Also, this method has got some drawbacks. The main one is, I think, that when you derivatize the protein by anything, you principally change its chemical properties. That means that it is not in native state, so it is very difficult to optimize the reaction conditions to stop the reaction in time when the protein is still as you wanted to study it and not an artifact of the entire procedure. Then you will have even maybe nicer peaks, and you will have more modifications, but they will be totally useless. Currently, the most popular modification of this method is based on treatment of the protein with hydrogen peroxide. It is rapidly irradiated by UV, and the amino acids that are on the surface of the protein are oxidated, usually methionine, cysteine, tryptophans. Unfortunately, it produces some rubbish from our point of view, and it is very difficult to interpret the resulting data. And the first method I would like to mention, I think you heard about it frequently on conferences or elsewhere, is protein chemical cross-linking. By some biofunctional reagents, you can connect reactive groups within the protein with a linker that has got distinct length, and in a correlation with other structural studies, it can give you information about the distance of the amino acids which were subjected to reaction of this linker. Usually on the conferences you see big professors showing you this last picture, like the nice result when they mixed protein and reagent and make a single MS analysis, they obtain this and they are finished. Unfortunately, the true stories are different, because every step of this derivatization or every parameter needs to be optimized, and it must be optimized individually for every combination of protein and the reagent, because if you leave it overreact, you then obtain a lot of crosslinks, but again, there will be on an artificial, denaturated protein. They will not reflect the native state. Also, the treatment of the MS data is not very easy because there are many kinds of peptides, many kinds of fragments, and in fact every linker needs another data treatment software. Nevertheless, after you obtain the result, then really it has got a high informative value, and the distance between the amino acid residues usually then correlates with other methods. It cannot be presented alone itself, because really this is not the absolute method, but usually it is now being introduced as one of the standard methods determining the protein structure. So in the past half an hour you could see how in different modes we can do even very rapidly the check of the dominant component of your protein sample. We can confirm its purity and identity, but also you could see that mass spectrometric tools, when they are used in a proper manner in combination with adequate techniques, can give you very deep insight in your protein sample and also can be directly used in structural biology. So that's all from me. Thank you for your attention.